Okay, we're going to talk about glucneogenesis. And this, of course, is switched on by glucagon. So during periods of starvation, glucagon is made in the pancreas. Target organ mostly is the cells of the liver, and it will switch on glucneogenesis. Now, we've got to realize that glucneogenesis, making glucose, is essentially going to be the reverse of glycolysis. So here we've got glycolysis, starting off with glucose, goes to glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, and then that splits into the 2,3 carbon units, glyceraldehyde 3-phosphate and so on. And then you get these final stages down to pyruvate, which lead to the production of ATP. And of course, pyruvate will then go into the mitochondria, into the Krebs cycle. So in glucneogenesis, we get various substrates, such as alanine, amino acid alanine, other amino acids as well, like serine, glutamate, and you get lactate and glycerol, and they can be turned into glucose, which by glucneogenesis, which can be released into the bloodstream. Okay, so let's see how this works and what glucagon has got to do with it. Right, so what I've done in glycolysis is all the steps which are reversible I've done with a red arrows, red reversible arrows. And you can see there are only 3 steps in glycolysis that are irreversible. So we've got the making glucose 6-phosphate, that one there, phosphorylating fructose 6-phosphate to form fructose 1,6-bisphosphate. And then, of course, the final step of all, phosphoenol pyruvate going to pyruvate, that is irreversible. So for glucneogenesis to occur, you need to make those steps go into reverse. So we'll talk about that, how that is achieved. You can't make that reaction itself with the same enzyme go backwards. If you do chemistry and you know about delta G, so these reactions with the green arrows, delta G is large and negative for them. So that means you can't go against it. They're hexagonic. Whereas all these ones with the red arrows, they are reversible. They're essentially equilibrium reactions. Right, so let's talk about this enzyme, pyruvate kinase. Now, glucagon will switch off pyruvate kinase. It will stop it working, which is really important because you don't want to be turning pyruvate back into glucose and at the same time making new pyruvate. You want it to be a sort of a kind of fruitless cycle of doing the same thing backwards and forwards. So that's switched off. Now the way that's switched off is because it gets the enzyme, it's phosphorylated by protein kinase A. And if you remember, protein kinase A, well, that's inactive unless you get cyclic AMP in the cytosol, and binding of glucagon to its receptor leads to the production of cyclic AMP. That's called a second messenger. That will switch on protein kinase A, sticks a phosphate onto pyruvate kinase, the enzyme, and that enzyme is then inactive. So that's good, you stopped glycolysis. Right. But that doesn't really explain how you make that step go backwards. And how to make that step go backwards is slightly indirect. So we have a look here. All right, what's going to happen is the pyruvate has got to go into the mitochondria. So I've drawn the mitochondria there. It's this red here. That's the matrix of the mitochondria inside there. Everything outside is the cytosol. So pyruvate goes into the mitochondria. Don't forget, pyruvate has got 3 carbons in it, 3 carbon unit, and you stick on a carbon dioxide and turn it into oxaloacetate, which has actually got 4 carbons. And the enzyme for that is called pyruvate carboxylase. Now, pyruvate carboxylase, if you want to make glucneogenesis occur, you've got to have an active pyruvate carboxylase. Now that isn't activated. that isn't activated as such by glucagon binding. But what glucagon binding will do is it will increase the rate of transcription. So you get more mRNA for that. And so you get more enzyme. And interestingly, cortisol, which is a glucocorticoid, cortisol, that will also switch on that enzyme, and that promotes glucneogenesis. And they both do that by making a binding factor, a transcription factor bind to upstream of the pyruvate gene if you've done control of gene expression. But glucagon will switch on gene expression. It doesn't affect the enzyme directly, but it will switch on its expression. Right. Pyruvate kinase, when you've got oxaloacetate with 4 carbons. Now, oxaloacetate cannot leave the mitochondria. It can't pass through the mitochondrial membrane. So it gets, also don't forget oxaloacetate is an intermediate of the Krebs cycle. Oxaloacetate is reduced to malate, another intermediate of the Krebs cycle. And then malate can pass out through the mitochondrial membrane. There's a specific protein transporter for it. It can then be reoxidized back to oxaloacetate. And finally, this enzyme here, phosphoenol pyruvate carboxykinase, that one, well, it will, again, oxaloacetate, 4 carbon unit, PEP, 3 carbon units, so you lose that carbon dioxide there. And then this can then go there. So you have made, by that long route, you've made the final step go backwards of final step of glycolysis. So for glucneogenesis, you need to have that going there. Phosphoenol pyruvate carboxykinase, well, that you may think, oh, would that be switched on by glucagon? Not entirely sure whether it is or not. You'd expect it would be, but there's a lot of work being done with PEP carboxykinase and also with pyruvate carboxylase, because obviously these are going to be very important in diabetes, particularly type 2 diabetes, where you're getting glucneogenesis, which shouldn't be occurring, and higher high blood glucose concentration. So there's a lot of research going on to those at the moment. Right, just have a look at those chemical formulae if you're interested here. Right, I've got it here. You have got that one there, that's pyruvate. This one here is oxaloacetate, the 4 carbon unit. You can see you've got another carbon on it there. And you notice that for this reaction to occur, you need ATP, needs to be hydrolyzed to make the reaction, make the reaction delta G, make it negative, so it will go forward. Now, PEP carboxykinase, well, first of all, it needs to get converted to this one here, malate. For it to be able to pass out of the cell, the oxaloacetate needs to go to malate. Malate can then be turned back into oxaloacetate, going backwards. You see it's a redox reaction. See, malate has got a hydroxide group there, whereas oxaloacetate's got a ketone, so you can see it's an oxidation step in that direction. And then oxaloacetate, PEP carboxykinase, it needs GTP as a source of phosphate, a high energy phosphate, and it's behaving the same way as ATP would, but it's GTP the enzyme requires. And there you've got the phosphate group stuck on phosphoenol pyruvate, which is that one there. Right, so that's that bit there. Now, we need to look, we know about the last step with pyruvate kinase. Let's have a look at these earlier steps, what's going on there. So this one here, this is the difficult one to understand, really. So you've got fructose 6-phosphate going to fructose 1,6-bisphosphate, and the enzyme that catalyzes that is phosphofructokinase 1. Now, that is an enzyme which is controlled allosterically. Right? If you remember what allosterically means, so we just look what's going on there. We can make that a bit bigger. Right, okay. Now, so we've got allosteric control, right? By fructose 2,6-bisphosphate. It's not fructose 1,6, slightly different. Phosphate is on a different group. Allosteric control means that an enzyme is either switched on or switched off by another substance binding to another site on the enzyme, not the active site, a different one that causes a conformational change in the enzyme. You can either switch it on, switch it off. Now, in this case, the allosteric moderator is fructose 2,6-bisphosphate, and when that binds, we'll do a little arrow up there. It switches on phosphofructokinase, and that is the main way in which glycolysis is controlled. Right, this is the key enzyme to controlling the rate of glycolysis, and it's very important. This fructose 2,6-bisphosphate intermediate controls it. So you've got fructose 2,6-bisphosphate bound, enzyme's really active. If it's not bound, then the enzyme's essentially switched off and glycolysis stops. So let's see, where does this fructose 2,6-bisphosphate come from? Well, it's made by this other enzyme, which is called PFK2, right, which is a very unusual enzyme. So why in a minute. But what fructose in that direction, it uses ATP, it will stick a phosphate on fructose 1-phosphate and turn it into fructose 2,6-bisphosphate. Right, now PFK2 is a pretty unusual enzyme because it's actually got two active sites on the same peptide, and they do exactly opposite things, which is pretty unusual. Right, one active site is the kinase activity. I've drawn that at the top there. That means it sticks a phosphate on to make fructose 2,6-bisphosphate. The other active site is this here, the phosphorylase activity. It cleaves off a phosphate group, the one on position number sorry, that actually should be fructose 6-phosphate there, not fructose 1, fructose 6, and that should be fructose 6. So it cleaves a phosphate off from position number 2 to turn it back into fructose 6-phosphate. Now, this enzyme PFK2 is crucial in determining the levels of the allosteric modulator molecule fructose 2,6-bisphosphate, and that is controlled by glucagon. What happens is, you can see here, PFK2 is phosphorylated by protein kinase A, mentioned before. Protein kinase A is only active when there's cyclic A and high levels of cyclic AMP in the cytosol, and the high levels of cyclic AMP only happen when you get glucagon or adrenaline actually binding to their cell surface receptors. So by having glucagon present, you essentially switch off the kinase activity, this one here, and you switch on the phosphorylase activity, so you get very low levels of fructose 2,6-bisphosphate. So, and that will stop that key enzyme working, which is absolutely essential if glucneogenesis is going to occur. So let's just have a look, go back to the glycolysis there. Yeah, no, oops, daisy. Right, so by having glucagon there, you've stopped this step. That no longer happens. And what will exist there, you've got fructose bisphosphatase, which is always present, and that can do the reverse reaction. So that means you can reverse this reaction, fructose 1,6-bisphosphate going to fructose 6-phosphate, and the forward reaction is blocked by glucagon. Right, the last reaction which needs to go backwards, this is glucose 6-phosphate, glucose 6-phosphatase. And that enzyme is it's not actually controlled by glucagon, but interestingly, it is controlled by insulin. Insulin will cause that enzyme to be, if there's any insulin present, it will cause it to be inactive. So you've got the, insulin promotes glycolysis, so you don't, and inhibits glucneogenesis. So if this enzyme is inactive, glucose 6-phosphatase, then you will not get any glucneogenesis. So pretty complicated kind of metabolic pathways there. Again, it is beyond the confines of the A-level specification, but just having an appreciation of it is pretty useful. And don't forget But you do need to know about the really essential role of protein kinase A here, which is of course activated by cyclic AMP. Protein kinase A will stick phosphates onto various different proteins and either activate them or inactivate them, depending on what they do. And that's really how glucagon has its effects in the cell.